all of the questions that you just answered were centered around the central theme of cells. And so obviously this class is called cell biology, so we're going to be talking a lot about cells, so it's important to understand what they actually are. So this definition I straight screenshotted right out of Google. So a cell is basically defined as the smallest functional unit of an organism. So essentially, it is the building block of life. So all life is made out of at least one or more cells. And so that's why it's really important to understand how cells actually work, why the components of cells couldn't survive on their own, but why a cell by itself can, and also how cells interact with one another to form more complex beings like humans. We're made out of like a lot of cells. When you like brush off the skin on the top of your arm or something, that's like billions of dead skin cells gone. So that just shows how many cells we actually are. And so it's important to understand their structure and stuff like that. Okay, so moving into introduction territory. So meet the instructor, that's me if you couldn't already tell. My name is Emily and I am a junior in high school from Maryland. And I have taught fossils, genetics, and just finished non-animal and nat for SAGE. I like biology, thankfully, or else I would not have fun here. I also do some art, I like to read webtoons, and also K-pop dance in my free time. So please don't roast me. Also, please don't roast me about my technology skills because I have a lot of trouble with that. I like cannot figure out Zoom for the life of me. So yeah. And then contact information if you ever want to reach me in any way, shape, or form. My Instagram is _mad, and then email, Discord. Probably the most direct communication you can have with me that would be like the most efficient would be Discord or Google Classroom, which you guys should all have joined, hopefully. Yeah. So say hi in the chat if you want. You can introduce yourself, maybe give a fun fact, or you can just nod because I know how awkward it is. Hi, Eleanor. I have seen you around a lot before. Yes. Meow, meow, meow. Can you translate that into English? And hi, Caleb. I am also familiar with you, a familiar face. Hi, Nikita. Is that how you pronounce your name? Give me correct pronunciation.
It's Nikita.
Nikita. Okay. If I mispronounce anyone's name, please just tell me. Also, if like your name is different from what your Zoom name is, because I know some people use their parents' accounts, just tell me what your actual name is. Because yeah. Hi, Alicia. Very nice to meet you. Hi, Claire. Hi, Angela. And I don't think you're human, Alvin, because humans don't meow. And hi, Alan. Hello, other humans. All right. Okay. That was very nice round of introductions. You guys can keep introducing yourselves if you want, but I'll move on to this side. So in the next few weeks, this is just an overview of the course, not gonna go over it or anything because you guys can read. So some general like housekeeping rules, always ask questions whenever they come up. And you can stop me whenever, tell me to slow down, ask me to explain something. Be nice to each other, please. We're just all trying to immerse ourselves in the wonderful realm of biology. And so for the games we play, names can be whatever, but should be appropriate. Also, you guys can definitely like chat during class. I don't mind that at all. and I will be monitoring it. Yeah. And have fun, but in SpongeBob meme lettering. I'm excited to be teaching this class, and yeah. So on to the actual boring lecture part. We might not get through everything today because I have like a lot of slides, since this whole topic is very like info heavy. So just like stop me at any time, and yeah. Okay, so first, to really understand cells, we need to understand what cell theory is and like where cells actually came from. So I guess the father of cells is Robert Hooke, who in 1665 was studying cork under a microscope. So can anyone tell me what cork is made out of? Or where cork comes from? If you don't know, you can also Google. Yes, okay, good. So cork comes from wood. So it was once a living thing. So underneath the microscope, he saw these tiny pores that were a part of the cork. And so there were like compartments, and he called them cells. And that's where we sort of get the modern concept of a cell, a building block. So the cell theory is just basically a set of rules and like widely accepted explanations that pertain to cells. So first, cells are the building blocks of life. So all living organisms are composed of one or more cells. 2, the cell is the basic unit of structure and organization in organisms. So that means that if you have different kinds of organ systems, so like you have the digestive system and also like the nervous system, they are all made out of special kinds of cells that play different roles. So the basic unit of structure is a cell, which determines bigger levels, which we'll look at in the next few slides. And also all cells come from pre-existing cells. So like no new cells are created. Now this leads to a debate, but before we move on to that slide, which is the next slide, I think. So Eleanor asked, are we cells? So we are made out of many cells, but we're not one cell. So like we have blood, and in blood there's like red blood cells, white blood cells. So there are individual cells within our body, and everything together forms a human. So we'll look at that in the slide. But okay, so the first concept, since all cells come from pre-existing cells, that kind of creates a question of where did the first cells come from if all cells came from pre-existing cells. So even though we're not really sure yet, so it's a running question in the field of biology, we have proven that organic compounds can be made from abiotic substances. So biotic things are living things, and abiotic things are not living things. And okay, so this isn't actually like Scioli cell bio prep. I will say that I did take inspiration from Scioli, but like for Scioli, I would say you want to dig a little bit deeper, because this is like the fundamentals, if that makes sense. So like use this as reinforcement, but not like guaranteeing that for at nationals. But yeah, so all life is made out of organic compounds. So an organic compound has the element carbon, and so inorganic compounds don't have carbon. So carbon is considered like the most important element of life. So you have carbon, hydrogen, oxygen, nitrogen, phosphate, and sulfur. Those are like the 6 elements that are the most abundant in life. But carbon is like all living things have carbon. And so all life has this common denominator of having the carbon element. So where did the first carbon actually come from? So in 1952, Stanley Miller, who is a scientist, obviously, he recreated atmospheric conditions similar to that of early Earth. So there wouldn't be much oxygen, there wouldn't be any organic compounds. And so because early Earth was like just forming, there would be a lot of lightning and also volcanic eruptions. And so his experiments found that under the similar conditions of the early Earth, if you had like a spark of electricity, this, after a lot of repetition, could actually create carbon-containing compounds. And so people hypothesize that this carbon-containing compound that resulted eventually formed into the building blocks of life. So this whole concept is called abiogenesis or abiogenesis, but it's probably abiogenesis, because it basically means that life is created from abiotic factors. And so this is sort of like a diagram of the materials he used. So first he had water in a small, like, I want to say that's like just a flask. And then so under it, he had a heat source. Now keep in mind that water is H2O, so it doesn't have C, it doesn't have carbon. So he would heat the water, and so it would turn into gas, water vapor. And then so this would lead to another tube where in this gas state, the water came into contact with an electric spark source, which mimicked lightning. And also in this chamber would be the other gases that would be found in early Earth. And then he would have a condenser, which would turn the water vapor and all of the other gases into rain. Because in the water cycle, you have precipitation, evaporation, and condensation, where gas turns into liquid. And so this liquid would now rain down, go into the water, and then the whole thing would repeat over and over again. And what he found was that after a few tries in the water, there was actually carbon. And so this kind of proved that, well, it didn't prove, but it provided great evidence that abiogenesis is a thing. Now, so some scientists also propose that it's the carbon-containing compounds were from like a meteor in outer space, which is also plausible, but those are just like postulates, or not postulates, hypotheses. Sorry. Okay, so Eleanor asked, are we cells? No, but we are made out of multiple cells that organize themselves based on various levels. So first, the basic structure and unit is a cell, but cells of similar types sort of congregate together to form things we know as tissues. So there are many different kinds of cells: red blood cells, nerve cells, skin cells, also cells that help you see color in your eyes. So each of them have a different function. And so these cells will come together and form tissues which perform a specific job. So for example, skeletal muscle, cardiac muscle, which is your heart, and things like that. So these are different kinds of tissues. And then these tissues will come together and form an organ. So for example, lungs, the heart, the brain, and then so these organs come together to form organ systems, digestive system, nervous system. You have the urinary system, skeletal system, muscular system, yeah. So, And then all of these organ systems come together to form a living being, an organism. So, Alan, what category do you think kidneys would fall under? Okay, good. So, urinary system has kidneys, which are an organ. And so, yeah, kidneys help filter your water, filter the waste from the liquid in your body, and so that becomes pee. Yes. Okay, that's cool. Yeah, I don't know. The urinary system is like pretty complicated to me because I can never remember like the placement in the body. Like I thought your kidneys were like near your legs for the longest time, and then I realized that's not right. Okay, so zooming out of the whole organism lens, let's go into a more focused point of view. So I'm not going to spend too much time on this slide. I mean, what do you want me to say? Like urine, nitrogenous waste, liquid waste? I don't know. Am I expected to not? Okay. Anyway, so all cells are made out of different kinds of atoms. Atoms are the smallest unit of like matter in our universe. There are different kinds of atoms that are each based on the atomic number. These different atoms are elements. And so, for example, H2O, 2 hydrogen atoms, 1 oxygen, atomic number of hydrogen is 1, oxygen is 8. Oh, you guys learned about atoms in Crime Busters. So, okay. If you guys learned any of this in Crime Busters, please just tell me to skip over it. Okay, so biological macromolecules. So this is going to be our actual focus for the class, which is kind of bad because we're already at 30 minutes. So I'm going to be talking pretty fast, but just tell me to slow down whenever, please. Okay, so atoms bond with one another to form 4 major kinds of biological macromolecules. So macro means big. So they are big molecules, not like micromolecules. So these 4 are nucleic acids, proteins, lipids, and carbohydrates, and they're each characterized by different things. And so their structure and their function are different. So one thing in common with all of these, except for lipids, but more on that later, is that they are polymers. So this means that they are made out of small repeating units called monomers, and these monomers are different for every group. So for example, if you have like a book and that was a polymer, then the monomer, the different repeating units of the book would be letters, because letters are repeated over and over again to form a book, a polymer. So yeah, chemical reactions and bonds. So different kinds of reactions help form these biological macromolecules. So the first kind of reaction that we have is a synthesis or condensation or dehydration reaction. All of these are like the same name. So essentially what happens is that you have these 2 monomers, and then so they each lose like, so one of them loses an H, the other one loses an OH, that forms H2O. So combined, they lose a water and then they bond to one another. The opposite is also called hydrolysis or decomposition reaction. A water goes in, gives them back their original structure, and the bond is broken. So many different kinds of bonds can be formed. So in the context of biology, covalent bonds are the strongest because everything in biology is studied in relation to water. So in water, a covalent bond is the strongest, and that's when electrons are shared. In ionic bonds, you have 2 ions, which are charged particles. So a positive will attract a negative, and the electrons are transferred. Hydrogen bonds are hydrogens binded to neighboring negative charges because the hydrogen is positive. So it's usually with a very negatively charged element like oxygen, nitrogen, fluorine. And then nitrogen phosphate fluorine. And then so van der Waals interactions are the weakest. They are random and based on electrons like zipping around. So they're not actually like because of some like charge or like valency. They are based on distance. So the order on the bottom is like strongest to weakest bonds. Also, another thing to note, covalent bonds, so compounds that are covalently bonded together are molecules. The other ones are just ionic bonds form salts, hydrogen bonds just form compounds, same with van der Waals. Okay, so functional groups. Oh god. Okay, you guys don't need to remember any of this at all, but just know that in biology, compounds are often found with like certain elements are found together a lot. And so these elements that are found together in the same position are called functional groups. And functional groups give the compound that they attach to a specific property. So for example, hydroxyl group, OH, adds polarity. A methyl group is just CH3, it's nonpolar because carbon-hydrogen bond is nonpolar, carbonyl is polar, carboxyl group is acidic and also polar. Amino group is positively charged, it's basic. Phosphate group is negatively charged, it's also acidic, and then sulfhedryl group is polar, also negatively charged. So in the terms of the pH scale, so pH measures the acidity of a substance. So it's 1 to 14, and then 7 is neutral. So yeah, pH 7 is neutral. So if I say something is acidic, then that means its pH is less than 7. If I say something is basic, then that means the pH is more than 7. And so acids are usually like sour, dissolve things. Bases are like bitter, and yeah. So Richard asks, what are the R's in diagrams? So R is just basically any compound. So it just shows like where the bonds would be attaching to. So for example, looking at the carbonyl group, let's look at the hydroxyl group. So oxygen can bind to two molecules, or not molecules, sorry, two compounds. So one of them is occupied by hydrogen, and another one can be attached to like anything. So to symbolize that anything, we use R. It's just like a sort of universal way to indicate that there's something that's going to be there, but it's not for sure. It doesn't have to be like one thing. It can just be whatever. Okay. Finally, into the actual macromolecules. Okay, so the first category is nucleic acids. So there are two main kinds of nucleic acids that we mostly talk about, DNA and RNA. So there are many differences between DNA and RNA, not just their names, but DNA, there are two DNA strands. So DNA looks like a ladder, while RNA is usually only single-stranded. Sorry, my voice is dying. Okay, so also DNA and RNA both have sugars in them, and so the sugar in DNA is called deoxyribose, and in RNA it's called ribose. Also for base pairs, you have thymine versus uracil, but more on that later. Hang on, I'm gonna get a drink of water. Sorry, was dying. And yes, Alan, that's completely correct. We'll look at it in like 2 or 3 slides, but yeah. So DNA basically provides the code for RNA, which makes proteins. So DNA stores genetic information. So let's say you're at a library and you have like the holy text of sage enclosed in a glass, like, do not touch container. And so in that container is like the book of the saints. And so that would be the cell's DNA. So obviously you're not going to go into the library and like walk out with this book. You have to make photocopies or like take notes or like take a photo or something. And that would be your RNA. So DNA has the original like codes for your genes, which determine your traits. And then RNA is basically a copy of the gene that leaves the nucleus where all the genetic information is and codes for proteins, just like Alan said. So nucleotides are the monomers that make up nucleic acids, the polymer. So A, C, G, T/U are the different nucleotides. So there are 5, but only 4 for DNA and 4 for RNA. In DNA, T is used, and in RNA, U is used. So again, thymine versus uracil. So the sequence of these nucleotides gives rise to a unique sequence that codes for proteins in the cell. We have more than 3.2 billion pairs of nucleotides in our genes, and I don't know about you, but I think that's a big number, and I wish to have that amount of money at some point in my life. So if you're wondering, like, why is it in pairs? Well, good question. So DNA is found in pairs because since it is double-stranded, it will make a ladder. So they have to pair with one another. So the nucleotide G pairs with C, and A pairs with T in DNA and U in RNA. So this is the structure of a DNA. Nucleotides are joined together by things called phosphodiester bonds. Okay, so this group is very dense. Proteins are the most functionally diverse and important group of the 4 biological macromolecules. Although you could argue that since DNA gives the code to RNA, which makes the proteins, DNA is actually the most important thing. But anyway, proteins account for 50% of the dry mass of humans. So even though humans are made of 60% water, of the other 40%, or like just the part that's not liquid, proteins make up 50% of it. So they have all sorts of function. They can store things like casein is a protein that stores milk. They are hormones, peptide hormones. They can aid in movement. Our skeletal muscles are made out of proteins, actin and myosin. Immunoglobulins or antibodies, they are proteins. Transport proteins, receptor proteins, and structural proteins. Like our hair is made out of a protein called keratin. Now, even though all of these are like good and well functions, the most important use of proteins, in my opinion, is for something called enzymatic reactions. So enzymes are proteins that speed up chemical reactions, and this is super important because our body is not sufficient enough to function on its own. We would make food too slowly if it weren't for enzymes. And so enzymes just basically help us be more efficient with our energy, and so that is important. So the monomers for proteins, also known as polypeptides, are amino acids. So there are 20 main amino acids commonly found, and so they have a basic structure, but then the only thing that differs from amino acid to amino acid is something called an R group, also known as a side chain. And this side chain can give amino acids different properties like nonpolar, negatively charged, acidic, etc. So different codons, or you saw the nucleotides earlier. So codons are a sequence of 3 nucleotides that form a specific amino acid. So for example, the amino acid methionine is AUG. That would be its codon, and it's made out of nucleotides. So peptide bonds also are a fancy name for covalent bonds in proteins. They're the ones that link amino acids through the synthesis reactions we just talked about. And there are 4 levels of protein folding. So I do not know why I didn't make protein folding the next slide. So going to this slide here, the primary structure of a protein is just a string of amino acid sequences. So the secondary structure, proteins start forming into patterns called motifs. And so these are a helix. These patterns are usually a helix or like a pleated sheet. Tertiary structure is when the protein, the different patterns start to interact with one another to give it its specific form. And quaternary structure is when multiple chains come together. So the 4th level is optional, and so these are the kinds of bonds that you would find in each structure. And then so proteins called chaperones actually help with the assembly of proteins, and this is just a cute little diagram if you want to read more into that, but I'm not going to because we don't have enough time. Okay, so going back to this again, enzymes super important. If you're gonna like take away one thing, just like remember that enzymes are super important. They lower the activation energy for a reaction. So for example, if it takes you 10 willpowers to do your math homework, enzymes would lower that 10 willpower to 2 willpower. So you have significantly more willpower to do other things because of enzymes. And so it just makes everything more efficient. And usually enzymes are highly specific, and so they have an active site that you can bind to. Let me see how much time we have. I'm probably not going to explain the, yeah. Okay, so homeostasis and regulation. So enzymes often undergo 2 types of chemical reaction feedback. So after a enzyme is like doing its thing, it'll usually receive feedback, which is like if you have a test and then the teacher gives you feedback and you know what to improve, what you did wrong, etc. obviously. But yeah, it basically helps regulate the entire process. So most of biology is based on something called negative feedback. And this is when the end product slows down the reaction. For example, if your body makes a lot of sugar in the blood, then the sugar levels goes up. The sugar in the body signifies to the body it doesn't need any more sugar because there's already a high level of sugar and the body stops making sugar. Positive feedback is destabilizing because you just keep going up and up and up. And so, for example, in birth, your body is pushing out the baby. The baby is like, hey, push me out more because it feels that it's already being pushed out. And so the mother's body keeps pushing the baby out until the baby is out. So that's an example of positive feedback. And so basically the reason why in biology there's so much negative feedback and not a lot of like this on and on and on positive feedback is because the body is trying to maintain homeostasis, which is the idea that it is in a constant state of like steady optimal living conditions. So if like I'm at a good body temperature, I don't want to like go cold all of a sudden. I just want to stay at this body temperature, and so I will do everything I can to keep me living in optimal conditions. So different factors that the body needs to consider: temperature, pH, negative, positive charge ions, and also salt and water balance, which is kidneys and urine and excreting waste. Okay, so this is just an example. I'm gonna skip over this. We already went over this. Central dogma. Okay, we should be able to get through this by speedrunner. So basically, the central dogma is the flow of information from DNA to RNA to protein. And so it goes through two processes called transcription, DNA to RNA, and translation, RNA to protein. So for example, how this whole thing works. Oh, also don't ask me why it's called the central dogma, because I have no idea. But that's just like the general concept name. For example, if you have a DNA that, since DNA has genetic material, a DNA gene codes for black hair. The RNA takes a copy of this DNA and leaves the place where DNA originally is. Then the RNA signifies to the other components of the cell what amino acids need to attach to the RNA so that it forms a string of amino acids, which forms a protein, and the string detaches from the RNA, goes into a chaperonin, and folds. And so this would fold into a hair protein that is black, which is how it works. So this is the codon chart, amino acids. I'm gonna skip over this. Okay, speed running this. Carbohydrates, third major group of biological macromolecules. The monomers are called monosaccharides, and the polymers are carbohydrates, also known as polysaccharides. So they are linked together through glycosidic linkages. So nucleotides were linked together to form nucleic acids through phosphodiester bonds. Proteins linked amino acids through peptide bonds. This is glycosidic bonds. So usually the formula for a carbohydrate is C, and then some number H2, the same number, and O, the same number. So for example, glucose is the most important and the most common carbohydrate that we use for energy, and the formula is C6H12O6. As you can see, 2 times 6 is 12, and then 6 and 6 are the same. And so it's usually classified through how many carbons it has. DNA and RNA, the sugar deoxyribose and ribose is a 5-carbon pentose. Cellular respiration depends on carbohydrate. Cellular respiration is what gives us energy. I'm gonna skip through all of this. Just know that the function of carbohydrates is to store short-term energy, in contrast with lipids, which is long-term energy, or fats. So it's technically not a polymer because there's not a lot of repeating units. It's just carbon and hydrogen, but it is non-polar because it does not mix with water, which is like the general property that all of lipids have. So there are 3 main kinds of lipids: fats, which is exactly what it sounds like. And so saturated fats are solid at room temperature and they don't have double bonds. Unsaturated fats have double bonds, which form these little like sort of bending, curved, not straight things, and so called kinks, and they are liquid at room temperature. Phospholipids are found in the cell membrane, which is also known as the phospholipid bilayer, which is what makes the cell membrane. Steroids are 4 fused rings, and they are used in hormones, cholesterol, and in cell membranes. These are just examples. I'll wait like 5 seconds, and now we will do a skincare. Whew, okay, that was a lot.